Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is A Digital PCR Liquid Biopsy Workflow for Mutation Detection and Therapeutic Monitoring. This webinar is sponsored by Stilla Technologies. Our speakers today are Dr. Danielle Enough, R&D Manager at ID Solutions, and Caroline Charkey, Vice President of Commercial Operations at Stilla Technologies. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of your webinar presentation. We will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets to enhance your webinar experience. So let me turn it over to Dr. Enough. Please go ahead with your presentation.
Hi. So good morning, everyone. So basically what I will talk to you today, it's how digital PCR can be powerful tools in the management of patients with cancer, especially with liquid biopsy, but not only. So the idea is to focus on the multiplexed, the complete workflow, and monitoring. So I will try to detail what we mean when we talk about complete workflow, why multiplexed is important, and how can we do with the Naica platforms to enhance diagnostics. So before starting, a few words about ID Solutions. So we are a French company based in the south of France, in Montpellier. So we are a small company composed of molecular biology specialists, and we developed assays and sell those assays, mainly focused on nucleic acid extractions and digital PCR analysis. So complete workflow. So our complete workflow is divided in, let's say, 3 to 4 steps. So the first one is the nucleic acid isolations. So in this case, cell-free nucleic acids. Then DNA quantification and qualification. And the last part where we use the multiplexing is the DNA genotyping. The idea of our workflow is to try to qualify extraction processes and to quantify each sample prior analysis and to use multiplex assay for mutation detection and genomic alterations. So if we look closer to the isolations, so we have solutions called ID extracts, which is used to isolate cell-free DNA. And the power of this assay is that it includes quality controls. So the first one, called the target positive controls, or TPC, has to be processed as a sample. So this control will allow you to validate reagents, the process of your isolations. The second one, the internal control of extractions, or ICE, has to be spiked in each sample. So we will try to focus a bit on this one because it will help you to quantify every samples. So the ICE is a small synthetic DNA from a non-human sequence. So the analysis of the internal control of extractions is a patented solution. I have also to mention that the internal control of extractions is also compatible with other extraction processes, not only with our ID extract solutions. So if we look to the target positive control, so the TPC, as I mentioned, you have to process this control as a sample, and it will allow you to validate your reagents, your instruments, the experimenters, the methods, the environments, everything going on when you try to isolate nucleic acids. So you can use the TPC as a sentinel, just make sure that each run of extractions perform well and that you have no loss of DNA due to reagents that was not well stored or anything else. If we look closer to the internal control of extractions, so this control has to be spiked in each samples. It will allow you to qualify your samples. So to detect the eyes, you will have to use the ID quant, which is a TACMAN-based PCR that works on either real-time PCR or digital PCR. This assay will allow you to amplify simultaneously cell-free DNA and the internal control of extractions. So the quantification of the cell-free DNA alone will allow you to calibrate your sample prior to further analysis. The quantification of the eyes will allow you to qualify your extractions and to make sure that your extraction was performed well for each samples. And if you use the eyes to normalize your cell-free DNA quantification, this will allow you to reduce bias during the process of nucleic acid isolations. So you will be able to normalize your concentration of DNA and to monitor your patient over time. Here's some examples of how the eyes works. So in the right panel, you have a graphic where you can see that 3 different concentrations of DNA were extracted 4 times for each concentrations. And as you can see in the red circle, you have a significant difference between the first replicates compared to the 3 other ones. However, if you use the eyes to normalize these concentrations, so in the left graphic, you will see that using the ratio of cell-free DNA over eyes, you have no more significant difference between each replicates. So this will really allow you to alleviate extraction bias and make sure that when you see a decrease or an increase of cell-free DNA from your patient over time, this is really due to the physiological states and not due to a bias during the extractions. Here's a small study that we performed on 5 patients where we measure the ratio of cell-free DNA over eyes pre and post surgery and for patients after relapse. So as you can see, post-surgery, you have a drop in cell-free DNA. And for the patient number 4, where we detect a relapse using imaging, we can detect a real increase in cell-free DNA for these patients. So you can make sure that what you see in your quantification of total cell-free DNA, it's really physiological and not due to an extraction bias. Now I'll try to go further into the multiplex digital PCR, but before going to the assay that we developed, I would like to make some reminders about digital PCRs. So the idea of digital PCR is to transform a one-way reaction of PCR into thousands PCR reactions. So you will partition your samples, making thousands of droplets. So for instance, between 25,000 to 30,000 droplets using the Naica systems. And then you will amplify your samples and measure how many droplets containing the target DNA you have. So using the number of positive droplets and the total number of droplets that you generated, using the Poisson statistic, you will be able to quantify with high certainty the concentration of your samples. So this is the basics of the digital PCR. If we go further into the multiplex digital PCR, to detect more targets, you will need to have more color detections or to use different strategies to increase the number of population that you can generate. So here's an example of the Naica systems where you partition your samples, you amplify your samples, then you will detect positive droplets in the blue channel, in the green channel, and in the red channel. So you can at least monitor 3 different populations in one wells. And the idea is to save samples and to monitor different targets at the same times. So the simultaneous detection and quantification of several genetic alteration in one PCR mixture will allow you to save your DNA while performing multiple testing. This requires appropriate design of the target probes labeled with a different fluorophores. The power of the digital PCR using the Naica systems is also that you can have the highest sensitivity. Here you have an example of samples with 50 microliter of DNA, which is the probability to pick up the red one. So this will depend on how many times you will pick up your samples with the low volumes, or how many times you can pick up your sample with the high volumes. So with our kits and using the Sapphire chips, the maximum sensitivity can be achieved with a triplicate assay. Indeed, we are able to use up to 15 microliter of DNA sample per tested well. So you can at least analyze all your sample in one well. So make sure that you have the highest sensitivity achievable. And using the multiplexing assay and the free color detection Naica systems, you will keep this maximum sensitivity and analyze multiple targets in the same reactions. So you can really be at the highest sensitive as you can and continue to analyze multiple targets. The other feature is that we are really easy to use. Indeed, our PCR mixtures are ready to use. So you only have to pipette up the 10 microliter of master mix, add your 15 microliter of DNA of all your positive controls, and then pipette these 25 microliter PCR mixtures in each well of the Sapphire chips. So you have only two pipetting steps to load your Sapphire chips. Few words concerning the Naica Crystal Digital PCR workflow. So as I mentioned, you have to load your chips with the 25 microliter of your PCR mixtures. So it will take you at least 5 minutes. Then you have to load your chip on the Sapphire geode. So the Sapphire geode will partition your sample and perform the thermocycling steps. So the first steps, you can partition up to 3 chips, so up to 20 samples per run, and you will end up with 25 to 30,000 partition per samples. And the second step is the classical PCR cycling conditions. The last step will be to remove your chips from the geode and to load the chip on the prism of the Naica systems to image all your droplet crystals and that make sure that your reactions perform well and try to detect the mutation that you want to see. And the last part will be the analysis of your samples. So basically, this can take you from 5 minutes to many times, depending on the complexity of the assay and the complexity of your samples. But you will use a really intuitive software when you can see your results in a 2D dot plots, in 1D dot plots, also in 3D dot plots. You can look at your droplet crystals, make sure that the positive droplets are not artifactuals. So you will see everything that happens during your PCR reactions. Okay, so now going back to the detection of mutation using these systems. So at ID Solutions, how we work to develop the digital PCR assay. So every design or every kit that we develop comes from a specific need from the clinicians. The idea is try to develop solutions that are really needed in the hospitals that have key features in for good diagnostics. So after this needs from clinician, we will design our primers and probes, optimize the assay using synthetic DNA, cell lines. When we are satisfied with the development, we will perform internal validations. So basically, internal validations are the determination of the limit of blanks at 95% confidence, the limit of detections. We will also perform some repeatability, reproducibility on the assay and on our positive controls. I have to mention that every assays that we developed come with positive controls to make sure that you really amplify every targets in your PCR reactions. The other features of our assay is that every assay works with the same PCR program. So you can use different assay in the same run. After this internal validation, we will send the assay to the clinician that asks for these solutions, and he will perform external validations on patient samples. So either FFPE, liquid biopsy, and we look at sensitivity, specificity, and we'll compare to the method that is using in the lab, so NGS, COBAS, RNA-seq, or other assays. Then we set up a vigilance system just to make sure that the kits is performing well, that the targets that we want to see are always key features, that if there is other targets to implement in the assay, if there is target to remove from the assay. This depends on how research goes and if there is new drugs on the markets. So every kits are optimized over time. Now, one question that was asked is what is the place of the digital PCR in the diagnostic labs? So here it's one example where the digital PCR has its place. So when you want to diagnose cancer, so let's see, for example, non-small cell lung cancer. So you will just look at if there is EGFR mutations. And if it's negative, you can use NGS to look at other targets. But as mainly the main mutation is the EGFR mutation, you will save time and save money. So in case of you need faster results, like if you have brain metastasis or any urgency, the digital PCR turnaround time is less than 1 day. So you can end up with fast results. Digital PCR can also be used to confirm NGS data, for example, for low abundance frequency. Then the usefulness of the digital PCR is for the monitoring of a patient, to monitor the treatment efficacy, for adapting the treatments. So you will use digital PCR to monitor your patients for resistance appearance, for example, if we look for the T719M in non-small cell lung cancer, or if you want to follow a mutation of your circulating tumoral DNA, for example, the L858R in non-small cell lung cancer, just to make sure that this mutation decreases over time, meaning that your treatment works. So if we continue with the EGFR, so we developed an assay called ID EGFR, when you can see in the free reactions almost every mutation that takes place in the EGFR. So sensitive mutations in exon 21 or exon 19, resistance mutation in the exon 20, so the T719M and the C797S. And for this mutation, we are able to discriminate between the cis and trans compared to the T719M. And the last reaction is for the rare mutations, for example, insertion in the exon 20, the S768I, and all the mutations in the exon 18, so the G719X. Now we'll show you some examples of how the results look like. So here's an example of the IDGFR for the sensitive mutation, so mutation in the exon 21 and 19. So on the right, you have a 3D projections of our positive controls. When you can see in the red population, it's the deletions of the exon 19. In the light blue, you have the EGFR wild type, and in red, you have the mutation in exon 21, so L858R and the L861Q. The small 3D projections show you a plasma which is wild type, so you only end up with the EGFR wild type population in light blue. On the left, you have the 2D dot plots of how the results look like, and you will see that it's quite easy to analyze. So you have the EGFR wild types in the blue-green populations and the deletion 19s, which is only green. And in the red channel, you can detect the mutation of the exon 21. So there is no doubt when you have to analyze this data, you just have to see if there is or not a populations. Here it's examples using FFPE samples. So the first samples was mutated in exon 21. So you can see that there is no droplets in the exon 19 population in green, and you only have droplets positive in red. The second samples was mutated in exon 19, and you can see that there is a population in the green triangle and no droplets positive in the exon 21. If you look at the reaction for resistance mutations, so in this one, you will end up with 4 populations in the positive control. So in the 3D projections in the right, you can see in blue, the EGFR wild type. In green, the C797S, which is in transpositions compared to the T719M. The T719M population, it's red, and the T719M with the C797S mutation in cis in yellow. So at the same, you have a plasma which is wild type, so you only have the EGFR wild type in blue. And on the 2D dot plot, you can see that it's quite easy to analyze. So in the first 2D dot plot, you can see that you have the exon 19 wild type, which is only blue. All the C797S population, which are only green. And if you look at the second dot plot, you can see that you can discriminate the 2 populations of the C797S, one which is only green and the other one which is green and red, so the yellow population, and you have the only T719M in red. So as the sensitive assay, really easy to analyze. Here is some example of what we got using cell-free DNA. So the first one was slightly mutated for the T719M, and the second one harbors C797S in cis. So as you can see on this 2D dot plot, so the T719M, we end up with 6 positive droplets. So the sample was not highly concentrated, but were able to detect the mutations and no droplet on the C797S. For the second one, we had 2 populations. So one which was only T719M positive, and the other one when we have a double positive droplets. So a part of the T719M mutation was associated with the C797S in cis. Regarding the ID EGFR for the rare mutations, so this one also it's quite easy to analyze. So you have the EGFR wild type in the light blue, insertion of the exon 20 in the dark blue, mutation on the exon 18 in green, so the G719X, and the point mutation in the exon 20, the S768I in red. So in the 2D dot plot, as previously, so really easy to analyze. You have your only green population, which are the mutation of the exon 18. The only blue mutation population, which is the insertion of the exon 20. And the only green population, which is the mutation S768I. So I will not show you data using this assay because we never had sample positive for those mutations. So we perform an external validation using this assay. So we compare both the COBAS and the NGS, and basically the concordance was quite high. We only detect 3 samples that were positive for the T719M that were not detected by the NGS, but for those samples, the abundance frequency was less than 1%. So this is quite normal. For all the other mutations we detect, like the COBAS or the NGS. We also developed other assays, so targeting the KRAS mutation on exon 2 and 3, NRAS for the same, exon 2 and 3, and the BRAF, where we detect mutations around the position V600s, and we are able to discriminate the V600E from all the other mutations. I will not show you how they look like, because this is quite the same thing than the EGFR. Now I want to show you another approaches that we use that was asked from the Professor Figueira-Labranger from the Hôpital de Marseille. So she's an pathologist, and she needed a brain tumor classification panel. So one for the adult cerebral tumors and the other one for the pediatric tumors. So we developed those two assays. So the adult cerebral tumor classification is composed by 4 assays, one targeting IDH1 and 2, one targeting mutation in the promoter of the HTERT genes, another one targeting the histone 3 F3A, and one for the amplification of EGFR. The other panel is composed of 3 assays. So one is the same for the adult, targeting the histone H3 F3A. And we have 2 different assays targeting FGFR1, where we will detect SNVs and the duplication of the genes, and the other one targeting BRAF, where we will detect the mutation V600E and the gene fusion with the KIAA. We also developed a PIK3CA assay that can be used in both panels. So I will focus on these last panels and show you how digital PCR takes place in the diagnostics of this kind of tumors. So a few words about pediatric cerebral tumors. So if you look at pediatric low-grade glial and glioneuronal tumors, this is the most common pediatric tumors. And it's a really heterogeneous group of tumors, so with a lot of overlapping histological criteria. So it's highly difficult in some cases to discriminate between the different types of tumors. So histopathologists need to look at genetic alteration, mainly on the MAP kinase pathway, to make sure that the characterization of the tumor is good, and this will help in the diagnostics. However, there is one issue, it's that there is not enough nucleic acids to perform all this analysis. As I mentioned, so those neoplasms are frequent, and they are often detected early. So the consequence is that there is little tissue available. So you have to choose between which alteration you have to focus on based on the histopathological analysis. So you have to decide if you want to use RNA for gene fusions or duplication, or DNA for SNVs. Indeed, you have different alterations that will be mainly present in different type of tumors, as alteration in FGFR1 are signature of dysembryoblastic neuroepithelial tumors, the V600E, which is signature of ganglioglial tumors, and the fusion of BRAF with KIAA, which is associated to pilocytic astrocytoma. So the request was to set up a multiplex digital PCR to detect all these recurrent mutations that are found in the glioneuronal tumors by using the same analytical chain. So by using DNA only extracted from small FFPE tissues. So they wanted to address both SNVs and duplication in one assay. So here is the example of how digital PCR can help in this kind of diagnostic. You can address multiple alterations quite easily. You don't need RNA to address some genomic alterations, so you can use a unique workflow. This is quite low cost, and as mentioned earlier, it's a short turnaround time. So now we'll show you some example of how this assay looks like. So here the example of the PIK3CA. So where you will see in yellow the wild type population of the exon 9 of the PIK3 genes, in green the mutation of the exon 9, and in blue mutation of the exon 20. And on the 2D dot plot, you can see that it's quite easy to analyze. So in the green and blue dot plot, you can see that you have all your exon 8 in green and mutation of the exon 20 in blue. And if you look at the green-red dot plot, you will see that your exon 9 can be divided in the wild-type populations, the yellow one, and the mutated population, only green population. Now, if we look now at the assay targeting FGFR1, so this one, we had to look at mutation in exon 12 and 40, as well as the internal tandem duplication of the genes. So the strategy that was used was to use the CNV approach by comparing the number of exon 8 that we can detect and the number of exon 16 that we can detect, because the exon 16 will be duplicated in these alterations. So here is the example. So if you look directly to the 2D dot plots, you will see that in the green-blue dot plot, you can compare the quantity of exon 8 and exon 16. So this will help you to address the internal tandem duplication of FGFR1. And in the green and red dot plot, you will see the mutation of exon 12 and 14 in the red population. For the BRAF assay, the same approach was used. So when you have the KRAS fusion with BRAF, you will end up with a duplication of this 3' part of the BRAF genes. So we will quantify the exon 3, which is not duplicated, and the exon 14, which is always duplicating when you have these gene fusions. So we use the same approach. You can see on the 2D dot plot that we will compare exon 3 and exon 14 to address the duplication of the 3' part of BRAF, which is signature of the gene fusions. And in red, you will see the mutation BRAF V600E. We also compare these results with reference techniques. So this assay was performed in the Hôpital Marseille. And the correlation was quite good, except for 2 samples that were mutated for FGFR1 using multiplex digital PCR and not using the reference techniques, and the same for BRAF. And those samples was with a low abundance frequency of mutation. So this highlight that digital PCR is quite sensitive compared to other techniques. So if I have to summarize this talk, what we propose at ID Solution with the Naica System platform is a complete workflow with an extraction process, which is quality control with internal control of extraction, positive controls, an assay to quantify and to qualify your samples using the internal control of extractions as a normalizer, and using the quantity of cell-free DNA to make sure that you have enough sample to perform your assay, and the identify that contain all the assays in the digital PCR multiplex kit that we developed. So I would like to say that the take-home messages are that multiplexing allow targeting multiple alterations while keeping sensitivity high, and that digital PCR is a good alternative to the only NGS way. So this helps you to pre-screen for main mutation before NGS analysis, and it's really useful when you have to monitor known mutations during treatment monitoring. Okay, thank you. Now I will let Caroline speaking.
Hello, everyone. So thank you, Daniel, for a very interesting talk. I just want to conclude by maybe highlighting our partnership that we've started almost 2 years ago with early on with ID Solution. So together our goal was really to have a complete workflow and a complete solution in order to address the clinician needs in the labs. So of course Stilla is a digital PCR manufacturer, and we did not have any kits available. So with this partnership and co-development and cross-selling approach, it will allow users worldwide to really have a complete solution for their whole workflow in oncology. So for those of you who are not very familiar with Stilla, so Stilla, we're also a French-based company. We started in 2013. We incorporated the company while actually Rémi Danglars, our CEO, while he was doing his PhD thesis at École Polytechnique, came across a new way of generating droplets within a microfluidic chip. And from there, he filed 3 different patents, and we officially started launching our Naica System platform in April of 2016. And since there, we have been adopted by more than 50, or now if we look at it today, it's more than 100 KOLs worldwide. So in Europe and Asia, and we are also opening a subsidiary in the Boston area. So we'll be direct in the US in the next few months. And in November of last week, we completed a 16 million euro Series A funding led by Illumina Ventures and other European funds. So now we have the resources and the means to really make digital PCR lab commodity and to continue innovating and collaborating with companies like ID Solution to really find new solutions for digital PCR. So I just wanted to address also a project that we're working on. So I don't know if you've seen recently, but we came up in Encore Target. There was a publication where we were the first ones to demonstrate 6-color multiplexing. And so we're going to be working with ID Solution also to have the kits that Danielle showed you in 3-color available as soon as possible in 6-color. But we're part of a Horizon 2020 project. We're led by Philips. So it's a 4-year EU-funded project. And the goal is really to combine liquid biopsy and MRI scans to predict and monitor cancer therapy response. And we're working on colorectal and breast cancer panels. And there's also Stilla involved, but Agena, DiaDx. And the goal is really to compare different techniques by mass spec, qPCR, and digital PCR. And so we're part of this initiative, and we're looking forward in being part of other liquid biopsy society initiatives. So we're continuously striving to be the first ones to give access to many more mutations and detection analysis. And finally, I don't know if some of you were aware, but in earlier this year in February, we were at the SLAS, the System Lab Automation Show in Washington, and we won the new product award for our new Opal chip. So this is a higher throughput version of our Sapphire chip, which will allow you to run 16 samples per chip. The input volume, Danielle was mentioning 25 microliters. So now we'll be working with an input volume of 8 or 7 microliters. We'll be able to perform 48 samples in 3 runs in 2.5 hours. And the droplet volume is 0.2 nanoliters, which is 73 microns. And we still have a really great dynamic range of 5 logs because within each chamber, even the footprint is the same. We were able to generate 20,000 droplets per well. So we maximize the surface of the chip, and this will be launched early in Q3 this year. So it's we're finalizing the beta tests, and we will be able to launch it early Q3. So these are just to give you a little heads up of all the innovation and R&D projects that are ongoing at Stilla, and we're currently growing. And thank you really for your kind attention. Thank you, Danielle. Thank you, GenomeWeb. And we'll be willing to pass back to Julia and see if there's any questions out there.
All right. Thank you, Caroline. Again, as a quick reminder, if you have a question, please type it into the Q&A panel. We'd also like to ask our attendees to take a brief moment after the webinar is ended to take our exit survey and give us your feedback. So with that, let us start with the Q&A. A question for Daniel. How long does it actually take to develop a new digital PCR assay? I think you had the workflow for that on slide 29.
That's quite difficult to answer how long it takes to develop. It depends on the complexity of the assays. Can take few months to up to 1 year, depending on how many targets you need, if you want to discriminate them, if it's only a screening assay. So, and this depends also on the sequence of the DNA. If it's like the third assay, which is IGC content, up to 80 to 100% of GC in the sequence, it's more difficult to develop. If you want a huge screen of the assay, for example, the KRAS assay, where we look at all the SNVs on exon 2 and 3, you have to validate every single variant and make sure that there is no different limit of detections between them to make sure that you can develop them and detect them, So basically, for easy assay to develop, it's a turnaround time of 3 months, and for some difficult assay, can be up to 1 year.
Okay, thank you. Another question for you, Danielle. Do you think it would be possible to adapt your assays to also identify epigenetic marks, in particular DNA methylation?
Yeah, for sure. This is the next step for us. It's try to develop assay to detect methylation defect or hypomethylation, hypermethylations. So yeah, for sure. The difficult part in epigenetics assays, it's mainly on the controlling the bisulfite assays and make sure that your reactions perform well before doing the digital PCR assay. So we will focus on quality control for the bisulfite before end up with the digital PCR assay.
Okay, thanks. Generally speaking, why did you choose the Naica platform instead of other digital PCR platforms out there?
The power of the Naica digital platform is that there is 3 color compared to the other company where mainly the viral systems that works quite fine, but you have only 2 colors, and Stilla it's really adopt, so and it's easier to analyze. You can develop more easily assays using this 3rd color detections. This is the key features for us. It's multiplexing, so 3 colors. It's easier to multiplex.
Yeah. Speaking of multiplexing, question for Caroline: what are the limits of multiplexing, and do you think you can improve it further?
What is? I'm sorry. The limit you mentioned. The limit of what?
The limits of multiplexing, and how can you improve multiplexing further?
Yes, so currently, as Danielle just mentioned, we were the first one to release a 3-color multiplexing. So it's really a key advantage of our platform. So you can use it. You can use the 3 channels for positive controls, for replicates, for negative controls. You could use it for mutation analysis to get multiple targets. But at Stilla, we're, as I mentioned earlier, we're really working really hard on getting our new 6-color instrument and reader to be released end of 2020. And so the power of multiplexing is really very, very important, especially in oncology and in translational research. And right now, what we did forget to mention is that all these kits are research use only, but some of them will be CIVD marked in the next couple of months or by the end of this year. And we are also working on an IVD platform by the end of this year as well. So right now it's really a complete solution for research use and translational research labs, but we will be getting into the IVD market as well.
Okay, thanks. That's great to confirm. We actually had one question that was asking whether the ID Solutions kits are CE-IVD marked, but I guess they are not at this point. They are research use only, correct?
Yeah, correct. So the idea is to see IVD mapped our assay. We need to release them to make sure that they work well, that they are repeatable, reproducible, and to generate data on real samples to make sure that the assay are correct before mark them with the CIVD.
Okay, thank you. Caroline, do you think if next-gen sequencing gets cheap enough, it will actually replace digital PCR, or do you think it will be staying for a while? Or the other way around, will digital PCR replace NGS?
Well, NGS is a really widely gold standard right now, and it's a very widely adopted methodology and technology. We think digital PCR is complementary to NGS, so it will not replace NGS until not now anyhow. So NGS is nice when you don't know what you're looking for. But once you know what you're looking for, digital PCR is much faster, it's cheaper, and in some of the cases, like Danielle mentioned, it's more sensitive. So when you have to give a patient reply and you have people waiting to be able to monitor and the patient and his treatment, digital PCR is really more powerful. So I think NGS won't be replaced by digital PCR anytime soon, but it's really a complementary approach.
Okay, thank you. Question for Danielle. What is the cost of developing a new multi-mutation assay, even though there might be a range, but can you just give an idea?
The cost for the developing of an assay? That's really difficult to answer because I have to take in account people that's working on the development. So I won't be able to answer to these questions because we are like 4 people working on the developments. We need samples. We have a lot of things to buy to the development, but the cost is really due to the people working on. So I can't answer to these questions. No, I don't really know, in fact.
Okay, fair enough. Question probably for either of you. Do you see any potential of digital PCR for non-oncology applications, for example, in infectious disease?
Yeah, of course. So right now this talk is focusing on oncology, and we see that in the digital PCR field, oncology publication is driving the field, but in terms of potential application in life science, it's sky's the limit for digital PCR. So we actually did a webinar not too long ago with Nature on infectious disease and MRSA. So we have people that are working on HIV. We have people working in NIPT. We have people working in organ transplant, in food testing, in environmental studies, in gene editing, in gene therapy. So, I mean, there's a really wide range of different applications, and also pharma and biotechs that want to do QC and dosage studies before producing their products. So, absolute quantification and endpoint PCR is what digital PCR does, and it really simplifies, and you can use it basically for any application in the life science industry.
Okay, thank you. Couple of technical questions for Daniel. Are you using for the DNA extraction kits that you have developed internally, or are you using a different vendor's extraction kit?
So we mainly use our solutions, but we also use other kits that are widely found in different labs, just to make sure that there is no bias due to the quality of the extractions. So yeah, we use all the kits, but mainly ours, because we need to generate samples for the developments. But all our assays are validated on different kind of extraction kits.
Okay, thanks. And also, what type of collection tubes do you use for cell-free DNA samples?
So in our lab to collect samples, we mainly use the Roche cell-free DNA collection tubes. We also work with EDTA tubes. We are not used to work with the BCT Strex tubes in France. I think now there is, it's more easy to have them, but it was quite difficult. But depending on the labs, and the country, they use different kind of tubes, and we try to validate all these tubes. So the EDTA, the Roche, and the BCT Strex.
Okay, thank you. One attendee says that a recent article showed that there were different results from two different companies. Specifically, these were Guardant Health and PGDx, when they analyzed the same liquid biopsy sample. Have you done a similar external validation comparison, and how do you ensure that your analyses are repeatable?
Yeah, that's good questions, because you have to identify which is right and which is wrong. So we, as I mentioned, so we always perform external validation using our assays and the reference techniques used in the labs. So if I take the example of the EGFR, where we found some T719M mutations that were not detected by the reference techniques. So if I remember well, so 2 were missed by the NGS assay, and 1 was missed by the COBAS assay. And for these samples, the same patient was monitored, and I think it was 2 months later that they used the plasma from these patients, and they detect the T719M. So this example confirmed that we were right and we detect the mutation before. But sometimes it's quite difficult to say if we are right or wrong. What I can be sure is that when you look at your digital PCR results, you can make sure that the droplets that you detect, it's a real signal and not something artifactual, because the mean fluorescence intensity of a droplet is caused by the efficacy of the PCR. So you are sure that you analyze a real mutation that is in the samples. Then maybe there were some either environmental contamination in this lab. There is many explanations. So the only idea is to correct the results that you obtain with clinical data and make sure that if you see some resistance, that the patient indeed regress or the drugs don't work. You have to use others' information that the only results obtained with this kit.
Okay, thank you. Question for Caroline. The new 6-color system, how long will the process time be for that?
The process time for the 6-color will be similar to the current 3-color. So right now, we do everything in 2.5 hours. The reading step only takes 10 to 12 minutes right now for the 12 samples. And we are developing the Opal chip that I mentioned that allows you to have higher throughput, so 48 samples per run with 3 different colors, and that the reading step is then a little bit longer, so it's 20 minutes instead of 10 to 12 minutes for 48 samples. So now if we go on a reader where we address 6-color multiplexing for 48 samples, the reading step itself will be between 45 minutes to an hour. So it will be a little bit more, but we're trying to reduce it as much as we can and to have it as closer and similar to the current 3-color reader.
Okay, thanks.
But it's currently in development, so we don't have the final times. But this is what we're aiming at.
Yeah. A couple of related questions, just a clarification. Does it take less time to run one sample compared to 12 samples?
Actually, if you run the instrument, you cannot run one sample. You have to run one chip, so that's minimal 4 samples. So you can run 1 chip or 2 chip or 3 chips on the current system. So whether you run 48 samples or 12 samples or 4 samples or 8 samples, the run time is essentially the same.
Okay, thanks.
So you have your pipetting step, as we mentioned, your preparing of your chip. Then you place it on the thermocycler, the geode. So it's about 15-20 minutes for the partitioning step, the droplet generation, and then after it takes in total the partitioning and PCR amplification takes 2 hours and 10 minutes. And then you transpose it into the reader, 10 to 12 minutes. So you really have a 2.5-hour turnaround time.
Great, thanks. Someone else remarks that 12 samples, what you can do right now, is kind of lower throughput than other digital PCR platforms, and is asking whether there are any batch effects when you run replicates across runs.
No, actually, currently, if you look at most of that's why we developed the Opal chip to address higher throughput. So our turnaround time is 2.5 hours, so it's pretty short compared to some of the other platforms there. So usually you can do, let's say, 96 samples per day. With our system, you can almost do this. With the Opal chip, you'll be able to do 4 runs or 5 runs during the day, so 48 samples times 4, or 48 samples times 5. With the current system, we can do 60 samples per day with 1 geode, but people that have higher throughput have multiple thermocyclers, so you can really increase also your throughput with the amount of thermocyclers you're using. So and the reproducibility from batch to batch is really the same. We compare if we run the same samples 5 times, 12 times, and the batch reproducibility is the same.
Okay, thank you. One more question for Daniel. It's about mutation monitoring, specifically in patients on TKI therapy. So the question is, if you not only look at T790M mutations, how many markers can you detect in one run? And do you have to use one panel or different ones?
So if you not only look at the T790M mutation, you will need to use a second panel. So basically, if you want to see the activating mutation, so an exon 21 or 19, so I would say at least 2 wells for a patient. So in one run, yes, as I mentioned, all our assays works with the same PCR program. So you can run EGFR, KRAS, and TEAT mutation with the same PCR program. So in this case of monitoring of patient with TKI, if you don't want to look only at the resistance mutation, you will at least need, for instance, 2 wells. So we are thinking if we can just change the EGFR assay, but for instance, it's easier if you have one well for the resistance mutation and the other one for the activating mutation.
Great, thank you so much. This is actually all the time we have for questions. So let me thank our speakers, Danielle Enough and Caroline Charkey, and our sponsor, Stilla Technologies. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you an archived version via email. So with that, thank you very much for attending this genome webinar.
Thank you.
Thank you.